morning students, we will continue carbohydrate metabolism. Today's competency is competency number 3.9 and topic is regulation of blood glucose level in fasting and fed state in normal persons and changes in the blood glucose level in diabetes mellitus. First we will see the regulation of blood glucose level in normal persons. Why it is necessary to regulate the blood glucose level? If the blood glucose level is not maintained in normal range, either it may increase above the normal range or it may decrease below the normal range. If the blood glucose level is increased above the normal range, it is called as hyperglycemia, which is harmful. Or if blood glucose level is decreased below the normal range, which is called as hypoglycemia, and it is fatal. Body has to maintain the blood glucose level in two different conditions. One is fasting state and another is fed state. It is also called as postprandial state or postmeal state. Now the body will maintain the blood glucose level in these two different conditions with the help of various hormones. First we will see the regulation of blood glucose level in fasting state. So what is fasting state? It is a state of a person who has not taken food for last 10 to 12 hours. That state is called as fasting state. And the normal blood glucose level in the fasting state ranges from 70 to 110 milligram per deciliter. In this state, if the blood glucose level is not maintained, then blood glucose tend to decrease below the normal range, which is called as hypoglycemia. And if the blood glucose level falls below 50 milligram per deciliter, then it is fatal for the body. So body tries to increase the blood glucose level in fasting state with the help of following hormones like glucagon, adrenaline, cortisol and growth hormone. These hormones stimulates and inhibits various processes in the body to maintain the blood glucose level or to increase the blood glucose level in this state. Out of this, the first mechanism is stimulation of glycogenolysis. The hormones glucagon and adrenaline stimulate the enzyme glycogen phosphorylase, which is the key enzyme of glycogenolysis, which causes breakdown of glycogen in liver and skeletal muscles. First we will see breakdown of glycogen in liver. When the glycogenolysis occurs in the liver, the end product of this glycogenolysis in liver is free glucose. So this is the pathway which shows the formation of glucose from glycogen in liver, and when this free glucose is formed, this free glucose travels through the cell membrane of liver or hepatocytes and enters into the blood. That means glycogenolysis in liver add glucose to the blood. But this is not the case in muscle glycogenolysis. In muscles, when the glycogen is broken down by the process of glycogenolysis, the end product is not glucose, but the end product is glucose 6-phosphate. This is because the enzyme glucose 6-phosphatase is not found in the muscle. So here the glucose 6-phosphatase is required to convert the glucose 6-phosphate into glucose, which is the last step of glycogenolysis. Due to absence of this enzyme in the skeletal muscles, the end product of glycogenolysis in the skeletal muscle is glucose 6-phosphate. Now this glucose 6-phosphate is a ester which cannot travel through the cell membrane of skeletal muscles to enter into the blood. Therefore, it will remain inside the cell only. And remaining in the cell, this glucose 6-phosphate undergoes a process of glycolysis. This glycolysis gives pyruvate, which may convert it into lactate, and this pyruvate or lactate then transported to liver and to form new glucose. That cycle is called as Cahill cycle or glucose alanine cycle, which is shown here. On the right side I have shown skeletal muscle where glycogen is broken down to its end product glucose 6 phosphate, which form either pyruvate or lactate. Then that pyruvate or lactate get transported to the liver. In the liver it may get converted into glucose by the process of gluconeogenesis, and now this glucose is added to the blood, and through the blood it may enter into the skeletal muscle. So this completes the glucose alanine cycle. And the second process which is stimulated by the hormones of fasting state is gluconeogenesis. The hormones like glucocorticoid, glucagon and adrenaline stimulates the process of gluconeogenesis by stimulation of key enzymes of gluconeogenesis like pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase and glucose 6-phosphatase. These fasting hormones not only stimulates the process of gluconeogenesis, but they also make available the substrate for gluconeogenesis, that is non-carbohydrate compounds required for glucose synthesis like lactate, glycerol, glucogenic amino acid and propionyl CoA. That means, so the fasting hormones stimulates gluconeogenesis by stimulation of its enzyme and making the substrate available for gluconeogenesis. How this substrate is available? First we will see lactate. Glucagon stimulates glycogenolysis in skeletal muscles. Just we have studied. The end product is glucose 6-phosphate, which may converted into pyruvate or lactate. This lactate is then transported to the liver and undergoes the process of gluconeogenesis. So in this way, glucagon make available a substrate lactate for gluconeogenesis. Adrenaline acts on adipose tissue, break the stored fat, that is triglyceride or triacyl glycerol in the adipose tissue to glycerol and free fatty acid. So in the adipose tissue, triglycerides are stored. Adrenaline break these triglycerides into free fatty acids and glycerol. The glycerol is then transported to liver, where enzyme glycerol kinase is present, which convert the glycerol into glycerol 3-phosphate. This glycerol 3-phosphate later on converted into glyceraldehyde 3-phosphate and glucose by the process of gluconeogenesis. Glucocorticoids or cortisol also provides very important substrate for gluconeogenesis, that is glucogenic amino acid. This cortisol or glucocorticoids acts on muscle proteins, leading to its breakdown to form amino acids. These amino acids undergoes transamination or deamination to form keto acids. These keto acids enters into the TCA cycle at various levels, which ultimately get converted into oxaloacetate, and then this oxaloacetate is used for gluconeogenesis. So this cycle shows various glucogenic amino acid enters into the TCA cycle at various levels. For example, histidine, arginine and proline undergoes transamination to form glutamic acid. This glutamic acid undergoes deamination to form alpha ketoglutarate and enters into the TCA cycle. Propionyl CoA is formed from valine, isoleucine and methionine, and this propionyl CoA undergoes carboxylation to form succinyl CoA and it enters into the TCA cycle. Similarly, phenylalanine and tyrosine are also converted into fumarate and enters into the TCA cycle. Aspartate undergoes transamination to form oxaloacetate. Similarly, alanine undergoes transamination to form pyruvate. So in this way, all these glucogenic amino acid get converted into keto acid and enters into the TCA cycle to form ultimately oxaloacetate, and that oxaloacetate is used for gluconeogenesis. Apart from stimulation of glycogenolysis and gluconeogenesis, the hormones of fasting state also tries to decrease the blood glucose utilization by the peripheral tissues by inhibiting the processes like glycogenesis, that is synthesis of new glycogen from glucose. They also inhibit the process of HMP shunt pathway and glycolysis in the peripheral tissues. So these are the 5 mechanisms by which the hormones of fasting state try to increase the blood glucose level. Now we will study the regulation of blood glucose level in post-meal state. First, what is post-meal state? It is a state which lasts up to 2 to 3 hours after meal. In this state, due to digestion of carbohydrates present in the food, in GIT, large amount of glucose is added into the blood. So blood glucose level tend to increase. If it is not controlled, then it may lead to hyperglycemia and loss of glucose into the urine. In normal persons, body will not allow the blood glucose to increase above 140 mg per deciliter. When blood glucose level start increasing, the beta cells of pancreas secrete a hormone called insulin. It is the only hormone which is secreted in the post-meal state and decreases the blood glucose level. Insulin decreases the blood glucose level by various mechanisms. The first and most important mechanism is transfer of glucose inside the cells. Insulin stimulates the enzyme translocase, also called as GLUT4 or glucose transporter 4, which are responsible for transfer of glucose from blood into cells or tissues. Here, insulin induces the movement of intracellular glucose transporter 4 molecules from intracellular compartment to the cell surface. That means insulin increases the number of GLUT4 molecules on the cell surface. When their number increases, they will transport more and more glucose from blood to tissues. That is, glucose uptake is increased. So this is the chart which shows various glucose transporters. Out of this, glucose transporter 4, it is present in skeletal muscles, adipose tissues and heart, and it is the only transporter which is under the control of insulin. In insulin deficiency, the number of glucose transporters on the cell surface get decreased, due to which glucose is not transported from blood to tissues. And these glucose transporter 4 are found on skeletal muscle and adipose tissues, which forms a bulk of our body and which utilizes large amount of glucose. These tissues, skeletal muscles and adipose tissues. The next process which is stimulated by insulin is stimulation of glycolysis inside the cells. Inside the cell, insulin stimulates the hormones of glycolysis like hexokinase, phosphofructokinase and pyruvate kinase, so that more and more glucose is utilized by the cells. Inside the cell, insulin also stimulates the process of glycogenesis by stimulating the key hormone of this pathway, that is glycogen synthase. Now this glycogen synthase converts the glucose into glycogen in liver and skeletal muscles. And all of us know that glycogen is a stored carbohydrate in the animals. So in this way, insulin try to decrease the blood glucose level. Also, insulin stimulates the pathway of HMP shunt, so that more and more glucose is utilized by this pathway by stimulating an enzyme called as glucose 6-phosphate dehydrogenase. Apart from stimulating the glucose utilizing pathway, insulin also inhibits the glucose forming pathways like glycogenolysis, gluconeogenesis and stop addition of glucose to the blood. If we carefully study, then we can find out that the pathways which are stimulated in fasting state get inhibited in post-meal state, and the pathways which are inhibited in fasting state get stimulated in the post-meal state by the hormones of fasting state and post-meal state. So this is about the regulation of blood glucose level in normal persons. Now we will see the changes in blood glucose level in diabetic persons or diabetes mellitus. All of us know that in diabetes mellitus there is insulin deficiency. When there is insulin deficiency, the antagonist hormones of insulin like adrenaline, glucagon, glucocorticoids, their level increases. The combined effect of this is increase in the blood glucose level due to following processes. First, inactivation of glucose transporter 4, inactivation of glycogenolysis, activation of gluconeogenesis, activation of glycogenolysis, and activation of hormone sensitive lipase. So we will see one by one these mechanisms. First mechanism is inactivation of various glucose transporters. The main glucose transporter which is inactivated due to deficiency of insulin is glucose transporter 4, which is present in skeletal muscles and adipose tissue. And as I have told you that these two tissues form large bulk of our body and they utilizes maximum glucose in the blood. Due to deficiency of insulin, the number of GLUT4 on the cell surface decreases, uptake of glucose also decreases, and hence blood glucose level increases in type 2 diabetes mellitus. So this is also called as insulin resistance. It is also found that entry of glucose into the skeletal muscles of diabetic person is half of the normal persons. Next is inhibition of glycogenesis. The insulin is required for the activation of enzyme phosphodiesterase. Now this phosphodiesterase enzyme convert the inactive glycogen synthase into its active form, and due to deficiency of phosphodiesterase enzyme, the glycogen synthase remains in inactive form, and hence glycogenesis, that is conversion of glucose to glycogen, does not occurs in diabetes persons. Second is activation of glycogenolysis. Again, the same enzyme phosphodiesterase which is stimulated by insulin, and due to deficiency of insulin, this phosphodiesterase formation get decrease. This enzyme phosphodiesterase is responsible for inactivation of enzymes of glycogenolysis. As this enzyme is absent, the enzymes of glycogenolysis get activated and the rate of glycogenolysis increases in diabetes mellitus. The next change occur in diabetes mellitus is activation of enzymes of gluconeogenesis. As we have studied, glucose inhibits the enzymes of gluconeogenesis and enzymes of fasting state stimulates the enzymes of gluconeogenesis. So these enzymes, pyruvate carboxylase, phosphoenol pyruvate carboxykylase, fructose 1,6 bisphosphatase and glucose 6-phosphatase get stimulated, and more glucose is formed from non-carbohydrate compounds in diabetes mellitus and added to the blood. The next mechanism is activation of hormone sensitive lipase. This enzyme is normally inhibited by insulin. In insulin deficiency, this enzyme hormone sensitive lipase get activated, which causes breakdown of stored fat, that is trialkyl glycerol, in the adipose tissue, to form glycerol and fatty acids. Both glycerol and fatty acids are transported to the liver. Fatty acids undergoes beta oxidation to form acetyl-CoA, and these acetyl-CoA in fasting state are utilized for synthesis of ketone bodies. So this is the mechanism of ketosis in diabetes mellitus, and the glycerol is used for gluconeogenesis. So these are some changes which occurs in diabetes mellitus which affects the blood glucose level. Finally, this is the WHO chart showing blood glucose level in normal persons, diabetic persons and impaired fasting glycemia. This completes the regulation of blood glucose level in normal persons and changes in the blood glucose level in diabetes persons. Thank you very much.